Welcome everyone to our 29th reading group. And it's our honor to have Professor Peijie Zhou here as our speaker today. So Peijie is a tenure-track assistant professor at the Center of Machine Learning and the Center of Quantitative Biology at Peking University. He earned his bachelor and PhD degree in computational mathematics from Peking University. From 2020 to 2023, he was a visiting assistant professor at UC Irvine, working with Professor Qing Mian. His research focuses on computational system biology, especially AI-driven modeling of complex biological systems using single-cell data, with work published in interdisciplinary journals like Nature Methods, Nature Communications, and PRX. So let's welcome, and it's all yours.
Okay, thank you very much for the very nice introduction and also for the invitation. It's a great honor to be here to introduce our recent work or some of some of work are still in progress about the general issue is we have multi-omics data and hopefully that's a time series one. How can we reconstruct the dynamics from it, right? So basically motivation is nowadays what is called the virtual cell. So basically, what is a virtual cell? Actually, different people have different opinions or definitions. So I think here is one um, that is actually a figure generated by ChatGPT, but actually I think it just tells a sense of what is a virtual cell. So basically, you have the inputs, no matter it's drug, different like molecules, or that is gene perturbation, the knockdown or knockouts of certain genes, and you really have a no matter it's black box or white box or gray box. So that's in silico digital twin of the cell. So that's the goal. And with this in silico cell, you could somehow predict the response of the cell, either under the drug response or what is the gene expression, or even what is the morphology, right? So after the perturbation. So basically, the idea is to speed up the drug discovery or speed up the basic biological research as long as we have a in silico digital twin of the real world cell. So you don't have to do a lot of experiment, or you could screen some candidates drug or candidate perturbation at first, and then to narrow down your search space, right? So basically, that's nowadays uh, what many scientists nowadays are working on, right? So that is a virtual cell. So just to let we can be more specific about the tasks of AIVC, especially we think that generative AI or generative machine learning models keep play a very important part because many of the important tasks of AI virtual cell are indeed the generative task. So let's just name a few. For example, uh, if you could do something like nowadays, given the text, you generate an image. But here, uh, in the cell experiment, as long as you have, let's say, some image, some about the morphology or histology of the cell, you could generate its molecule. For example, the gene expression or the protein abundance. So basically, that's one very important application of the virtual cell, right? So you have the cross-modality argumentation or imputation, so that you could really do some very low-cost measurement. For example, that's a take image of the cell, and you can use the AI models to automatically generate other modalities, especially some expensive measurements, just like omics data, right? So definitely that's one possible or one important application of the virtual cell. So the other thing I think also many people are working on is something called perturbation. So basically is after the treatment, after the gene knockdown, gene editing, you have one population and you just want to predict what if a new drug or a new perturbation comes and what if you generalize to new cell line or cell type or similar kind of things, right? So basically, that's a perturbation prediction task or generalization task. So of course, some very important works like a cell flow or like multi-marginal formation have been addressed this. There are many works still come out. So the last thing I think also the focus of my talk today is another very important task in the AIVC is you want to do some spatial temporal reconstruction. So basic idea is nowadays, although we have very high resolution omics measurements, no matter it's single cell or nowadays we have spatial, even you can cut into different slices and reconstruct a 3D. For example, that's a 3D fruit fly embryo. But what happens for the sequencing data is by nature, that's destructive measurements, right? So you can have a very rich information about snapshots, about even the physical location of the cells, as well as the gene expression, the molecular feature of all the cells. But that's a static snapshot. And it's possible you take, for example, the embryo at different stages, but unfortunately, you can't measure the same embryo. Or in the single cell measurements, you cannot measure the same cell because the cell are killed after you're doing the sequencing, right? So basically, you can now have continuous trajectory. So here, the very important thing, just using a similar model in the machine learning or computer vision, is you just observe some snapshots or some static images. Could you interpolate or extrapolate in the future in terms of the temporal points so that you can construct a video based on that, right? So basically, that's called something called a spatial temporal reconstruction problem. That's also very important to realize AIVC in the omics data. So imagine later you can only measure very sparse uh, very sparsely in the time resolution, but you can recover continuous dynamics. So that's also very practical usage in the no matter that's basic research or translational study, right? So here the issue is we want to do something really make the AI virtual cell really dynamic. So basically that's from the very well-known review. So actually what AIVC want to do is to realize a virtual instrument can predict the function, behavior, or dynamics of the single cells. On the other hand, the key issue, or even we say that's a bottleneck, is we only got the snapshots of the omics measurements. So how can we combine some dynamic models, which is a very natural generative model, because with actually with some like physical laws or some modelings, you could predict or interpolate any points you like because that's a rule-based system. But of course, we want to combine with data to generate the continuous dynamics underlying the measurement, right? So basically, that's the uh, that's the background or the motivation of our research. So just let's be more specific on the technical side. So what kind of the candidate theory that we could use? So definitely one very appealing approach or candidates in mathematics that is called optimal transport. Basically, it could handle the change of the distributions, right? So you have one distribution, then you have another. The optimal transport just want to find a way that is most efficient or least costly to transport one distribution to another. So why that's important for the omics data, especially the dynamical modeling, simply because based on the snapshots, you don't have the paired cells, but actually the optimal transport naturally define a coupling or a most probable coupling between the cells across the snapshots. So that's really provided to make something almost impossible to be possible to infer the dynamical change of the cell states, right? So basically, we have already seen a lot of very great applications of the optimal transport in the multi-omics data models. For example, that's one in 2019 published in a Cell that's called Waddington OT. Basically, use a modified control form to couple the cells sequenced at different time points, right? For example, there's an iPSC differentiation data. So you could just by defining an OT coupling, you could possibly define something like a Markov chain is given the current state, what is the probability that the current cell goes to another state in another time point, right? So basically, that's a dynamical system could be recovered from the static measurements. So another very important application of OT beyond spatial temporal is what we called in the perturbation task. For example, the cell OT. So we just people can just parameterize a Monge map. So basically, that's before and after. So you only have the distributions, but you can parameterize a mapping to couple the different distributions so that given a new population of the cell, you could generalize to predict what is the response of the current cell or unseen cell population, right? So basically, that's also very natural but very clever application of the optimal transport. Another thing, maybe that's also very, very well known, that is the Mascot model. So basically, that's a very, very comprehensive uh, method or actually a software package. Uh, the basic idea is they could do something called the multi-model integration, right? For example, you have ataxic measurements, you have RNA-seq measurements, you have proteomics measurements. Basically, they could be in the different space. Or, for example, for the spatial temporal data, you have two embryos that are sampled that are in the different stage. So the key idea in the mathematics is they are not in the matrix, not in the same metric space, right? Imagine the space of the protein might not be the same with the space of, let's say, morphology or even mRNA, right? Their abundance or their distribution are actually very different. So here the key idea is you just define the metric within the same space, and you have some uh, metric preserving or similarity structure preserving transportation, right? So basically, that's the idea of GWOT or Gromov-Wasserstein OT. So it could handle the cross modality integration or mapping very well, right? So basically, that's another important application of OT. So maybe one is very curious. So the OT is already very great. They can do a lot of things in kind of the different things. So naturally, how could we apply optimal transport in the dynamical virtual cell, or actually the dynamical reconstruction of the omics data? So basically, I think here the idea is optimal provides a very, very powerful mathematical tool. But actually, in the real biological transport, there are a lot of details that we also need to incorporate into such process, right? For example, many OTR has a static form, but actually what's important is very good to know the continuous dynamics between the observed time points, right? Because the biology process is actually intrinsically that is a continuous or dynamical in time. And also a very important thing that is also actually uh, handled very well in the static form, for example, in Moscow or in the Waddington OT, is actually it's not a very straightforward coupling between probability distributions, but rather that's a coupling between arbitrary measures, because the cells can proliferate or the cells can undergo the apoptosis, right? So actually there's a change of the cell number, especially if you see a spatial omics data, right? For example, that's during the mouse embryo development. Actually, the cell number changes a lot. So how to better cope with that? That's an unbalanced issue that we need to consider to make the model align better with the real biology. And of course, the biological process, especially in terms of gene expression or state transitions, there are a lot of intrinsic noise as well as extrinsic noise, right? So how to model the noise, the stochastic effects, the stochastic dynamics is also very important. And also, there are a lot of database, for example, about gene regulation or even multimodality. For example, ATAC gives a lot of information about gene regulation, how to integrate this prior knowledge into the optimal transport that to make more constraints about the process. That's also very important, and especially for spatial omics data, how to model the spatial migrations. And also, lastly, it becomes increasingly important is the majority of the optimal trans models just to think the cells are independent when just to model the transport, but actually we know that cell communication, the cell interaction is very, very important during the no matter it's in the development process or in the disease process, right? So how to better model interacting particle system during the optimal transport. That's another very important issue. So basically, that's an incomplete list. There are a lot of the biological priors or many interesting biological problems or phenomena that we need to model, uh, but under the common, the big framework of optimal transport. So there are a lot of opportunities based on the mathematical model or deep learning algorithms that we could do, right? So basically, we just want to begin with a you can call it first principle, but actually it's not, right? So basically, we want to understand how to model the spatial temporal omics problem. So basically, is you could consider what you observe the samples from the multi-omics measurements are the realizations or some samples at different time points. But they are sampled from a continuous distribution or not necessarily probability distribution because it's now normalized. It's not normalized, but actually it could change with time or even grow with the time, right? That's possible. But anyway, there's a underlying distribution that is changing with time, and actually the state is you can either it's gene expression or you can also record spatial location. So basically, that's the first assumption, right? So basically, there's an underlying uh, probability distribution that I want to do. That's also very uh, nowadays very common to the generative model in the machine learning, right? You observe samples, but it will somehow want to get the information about the underlying distribution. That's the first thing. The other thing why we call it has some principle is because in the dynamical system models or computational systems biology, people tends to have some form of the equations to model the change of the distribution with respect to time. For example, that's a Fokker-Planck like equation. You have the driving force that is drift, but you also have the stochasticity that is a diffusion. At the same time, you can have the same kind of source that is a growth, and you can add more. For example, the interactions that I will talk later, right? So basically, it's a very general formulation of the problem, but instead of assuming any possible distribution, the change of the distribution already have some constraints in terms of its structure, right? So basically, this PDE specify a structure, but actually this term, for example, what is the change rate of the in terms of the gene expression? What is the change rate of the spatial location or the spatial migration? They are unknown or to be determined from data, right? We only have a rough picture or very rough constraints of the format of the PDE model, of the change of the distribution, but there are still a lot of things to be determined from the data. Or from mathematical perspective, you could consider in this way, you have some point clouds, right? They are actually evolving with the time, but actually there's a continuous manifold underlying the data that is also changing with time, but actually this change of the manifold could be described by some partial differential equations. But on the other hand, how can you just based on this discrete measurements, you just recover the temporal evolution law of this point clouds, right? So basically, that's the abstraction of the spatial temporal multi-omics model problem. So, um, so actually, just based on this, there are also a lot of very great previous work or relevant work. For example, one very well-known or very important model is the static optimal transport. Indeed, has a fluid mechanics or just a PDE or dynamical formulation that is called a Brownian form, right? So basically, it just tells so how to model the optimal transport from to this time is probability distribution because you assume the conservation of mass. Although later we will just relax these constraints, but let's just assume there is the conservation of the mass. So basically, that is continuity equation, right? Just to describe the transportation process. So we have two distributions at two time points or consecutive time points. So what the optimal transport want to do, or actually that's actually equivalent to the static Kantorovich or Monge form, is actually you minimize from all the possible transport PDEs that can connect two distributions they are observed, you find one with minimum kinetic energy, right? Because that is actually v squared times the mass, or actually that's the same units with the kinetic energy, but you just take integral over the space and over the time. That's accumulative energy. But anyway, there's a very elegant physical interpretation of the Wasserson distance or optimum transport is you want to find the least energy way of transporting the mass, right? And actually, that's equivalent to the Kontranovich OD form. And actually, for example, Alexander Thom, he just his group was just propose a uh, uh, very first deep learning algorithm to solve that based on the continuous normalizing flow is you just parameterize because you just want to solve a constraint optimization problem to find the vector field P, right? After have the P, you just recover all the dynamics of the cell in this constraint. So you just think uh, uh, this is a vector field, and you use a neural network to parameterize that. But actually, each particle obeys the characteristic line is ordinary differential equations. So you could use a neural ODE solver to optimize this P, right? So basically, that's called a trajectory night. That's a very important work in this direction. So another kind of issue is now how to modify this equation. How about putting more stochastic effect into that? So basically, that's called a Schrödinger bridge problem. That's also very classical problem in physics. So actually, Schrödinger proposed this problem is you just imagine you have a prior about the system. You just think this is Brownian motion or Wiener process connecting the two distributions. But actually, it happens the true process may not obey that, right? So actually, in terms of probability, there's something called a large deviation theory just to quantify how far is your observation from the prior assumption, right? So basically, the idea is you have, for example, the x, this random variables are sampled from the distribution, we call it p, but actually you have an empirical distribution because you only have the finite sample, and you can do, for example, the statistics to estimate empirical distribution, and you want to ask what is the most probable, or actually you can quantify what is the distribution of empirical distribution, right? So it's the high level is in the level in the space of the distribution. What is the distribution of empirical distribution? Actually, that's called the sum of large deviation principle. Just tell you, okay, it is relative. It has actually it has an exponential form, and the there's something called rate function that is a KL divergence between the the empirical distribution and the prior distribution, right? So basically, that's a very general formula about empirical distribution. So now go back to the Schrodinger's question: is you have prior knowledge that's called YT, is something like a pure uh, uh, the heat, the heat movement driven, right? So basically, that's a free particle, and but actually you observe something it's called xt, that is empirical process, and you really want to find a process that is most probable based on the large deviation principle, but actually this time is in the stochastic process space. It's actually for the prior knowledge process, it has a measurement, we call it y, and you want to find all the from all the possible movements, the stochastic differential equation, the particle, the laws that particles can obey. You find the one that minimize the KL divergence, but in the again, it's in the path space of the X process with respect to Y, and you want to find the minimum KL divergence is actually the most probable one or the least action one, right? So actually, this form, many people already have the great work have shown that is actually equivalent to the entropy regularized optimum transport. So just to have an entropy term to penalize that. So actually, that's also very important. For example, inspire the Synchron algorithm to solve the static OT problem. So, but actually, this still seems a little bit abstract, right? What is the KL divergence in the path space or stochastic process space? But actually, the idea is you could do some measure transformation using the basic stochastic process formula, but actually that's already a known results. Actually, we just provide a intuitive, intuitive interpretation, or just to show how to derive that is just based on the Garrison-Of transformation of the past, the change of the measure. But in the stochastic process, you could, in the end, after some derivation, the idea is you can have a very similar form with dynamic OT. Is this time you have a Fokker-Planck equation constraint, the one with the diffusion, but you also want to minimize some kind of the generalized kinetic energy of that equation, right? So basically, that's the formula, the equivalent formulation of Schrödinger bridge, but this form is more physical or connected with the dynamic optimum transport form, right? So this is the stochastic process. How can we treat that? So another issue is the unbalanced issue, because in static OT, for example, in Waddington OT or Moscott, they could consider the unbalanced issue by formulating an unbalanced OT problem. But how to formulate in a dynamical way? Basically, that's called a Waddington Fisher-Rao metric. But here the idea is simple: is you just assume there could be the creation of the new cells or the death of the cells, right? So basically, you just in the PDE just governing the probability, not probability, but the distribution of the cells. There's another sink term or source term. So basically, the G is the growth rate of each cell. It can depend on the cell state and also depends on time t, right? You just assume this is a new family of the PD equation that you want to reconstruct from the data given the observations at some snapshots. And you here the Waddington Fisher-Rao said, okay, beyond only minimize the total kinetic energy, actually there is a regularization term for the G, right? You just expect if there's no prior knowledge, we just expect from all the possible dynamics that explain the observations, you just want to find the one with the as small as the growth rate is possible, right? So just to keep your dynamics simple. So the idea is quite straightforward, but actually this WFR form has very nice properties. For example, it has some gradient flow structures that could guarantee the the convergence algorithm. So, but here the issue is now you have a problem, but how to solve that using the machine learning, right? So because here the velocity as well as the growth need to be determined from the data. It's an optimization problem, but it's high dimensional. It's also PDE constraint. So how to solve this? The key idea, very similar to the trajectory net, is we want to formulate a particle system using the Monte Carlo approach to approximate this PDE. But actually, the idea is you just follow the characteristic line of this PDE that actually is the dynamic of each cell or each particle, but the contribution of the growth just to make it a weighted particle system. So using this approach, you don't have to really split the cells during the simulation because the splitting process is discrete. And in other words, in the training, you cannot backpropagate. But you use a weighted particle approach. Each particle, G is a neural network, and P, the velocity, is a neural network. So you just simulate the particles. The particle, when they move, they can just carry the parameters, the weights of the neural network to along the characteristic line to the observed time points, right? So you have a point class. Imagine that's the virtual particles. They carry the neural network parameters. But at the observed time, you have some real particle, right? The key idea is you just make a penalty. You want the distribution of the virtual particle satisfy match the distribution of the real particle. At the same time, you want to minimize energy, but energy you can also express the whole ensemble mean as the temporal mean along the temporal integral for each particle. So basically, it becomes a very standard neural ODE problem. So you can use the backpropagation to optimize both P and G, right? So basically, that's the idea of using Waddington Fisher-Rho metric to do that. So now just comes what do we the title of the talk today is what if we want to model both. You both have stochastic and you have unbalanced. How to do that? The key idea in mathematics is you want to transform a unknown problem or new problem to some problem that we have already solved. For example, the WFR is something we already solved. So the key idea is now you have Fokker-Planck equation constraint, how to transform that into the original, the one without a diffusion term. So here the idea is also very common in the diffusion model that is proposed by Yang Song, now in OpenAI. Is the key idea is you just do something called a probability flow transformation, so that you can transform a Fokker-Planck equation to the Liouville equation of the ODE, right? So basically you just define a score function, and if you use the b minus the score term, defined as a new velocity field, and the ODE of the new velocity field v, the associated Liouville equation, or the PDE, the distribution of this particle system, is the same with the original Fokker-Planck or the stochastic differential equation system, right? So here's the idea. So you just make a change of variable. So after some calculation, you can find that you just there are some new term in the energy function, right? There are some, for example, one term is called the score function term, and also there are some cross term here, right? So based on that, you can naturally do something like, but here the constraints PDE now becomes the something we already solved, right? So basically, that's what we saw in the WFR. So we can use the weighted particle to solve that very efficiently instead of also using instead of using a SDE solver, you can use a ODE solver to efficiently solve this problem. So here the idea is now we just design different laws. It's still a weighted particle system. So basically, first we have a transport energy. So that's the same with the original one, but you have more terms, especially you have the term with log p. So you need another neural network to parameterize that. That's called a score matching. And another thing is what we said. So how to make the particle system match with the true multi-omics data or spatial omics data is you just evolve the particle system. And at observed time points, you have a loss. You make the mass match first, and also the distribution, the shape also need to match, right? So we did that two terms here to match the observed time, the particles at observed time points to make sure that your the PD system can model the data. The last one is the pin is actually for score matching too, because here we have log p. Just because we use the trick to transform the RUOT problem to the original to the WFR problem, right? So basically, you have this term, and you have three networks to do that, and you just design the backpropagation to solve that. So of course, the solution is not easy, to be honest, because you have three networks you want to train, and you have like here we have three laws, right? It's a multi-objective training, so definitely the convergence is a very challenging issue. So here in what we find empirically is we can use some pre-training techniques so that you could somehow use the flow matching to give an initial solution, and based on the initial solution, you can just start training the true objectives so it could converge more stably. So here there are some results. For example, this is synthetic data. Is we just create a system. For example, here just to show the importance of modeling the unbalanced effects. For example, here is a quiescent cell that never change with respect to time, right? Here at this corner. And you also have a growing cell population. At the same time, it's also changing state, right? So assume at time zero, there's the initial population of this population's here. Then with respect to time change, it will move in the gene space, but also grow. So if you just use a balanced or original Schrödinger bridge to model that, because you just normalize, although this, the quiescent cell, they never change, but the proportion of the growing cell population will dominate. So the static OT or the balanced Schrödinger bridge problem will tend to give a solution that the quiescent cell will transit to the growing cell population, but actually that's not a true dynamics, right? But here, if you just explicitly model the unbalanced or the cell growth effect in the equation, you could somehow get rid of more reasonable solution in terms of the true dynamics, right? So basically, that's what we learn about the importance of unbalanced effects in the data. Another thing is we test the scalability.
I have a question.
Oh, sorry. Okay. Yeah.
Just to be clear on
Okay.
what does the growth rate means here exactly.
Uh-huh.
It is like new cells that are created upon division, or is it like a growth rate in cell size or cell feature value? How do you here exactly
Yeah.
define the growth rate?
Okay. Yeah. Thank you very much. So that's a very great question. So basically, here is the cell division. That's about cell number. So for example, you see here this equation. This row is actually the distribution of the cell. So we have
Yeah.
G row with
Yeah.
respect to time t. So that's the cell number growth. Just for example,
Okay.
here in embryo data, there are more cells during the development. Exactly.
Yeah.
Yeah. So that's the growth rate of cell.
So because there are more cells, you like have if you do simple OT computation, you'd have a map from like let's say 1,000 cells
Yes.
in the source and 2,000
Yes.
cells in the target, for instance.
Yes.
Okay.
Yes. Yeah. Exactly. So that's assumption of
Okay.
balanced OT. Yes.
Yeah.
Exactly. But
Yeah.
actually, a lot of data, especially development data or even nowadays perturbation data, actually there's the unbalanced issue that maybe we need to consider in terms of real biology. Yeah. So basically,
Yeah.
that's the point. Yeah.
Sure. Okay. Thank you.
Okay. Thank you. So
Oh
any
no,
other questions, by the
and
way, so far? Okay.
one more,
Yeah.
yeah,
Uh-huh.
one more
Okay.
question. Did you try that on a real data? Because here it's a synthetic data. But...
Exactly. Yeah. So
Okay. Thank
this is the real
you.
data. So that's from the blood development. So we have three measurements. So basically, because this algorithm could also infer the growth rate. So actually, you can find at the beginning that is the stem cell, the blood stem cell. Actually, they have the larger growth rate, and then afterwards they have the just to give three snapshots, we can reconstruct the the stochastic dynamics. And actually, in the paper, we also did some quantitative uh, uh, benchmarking. For example, you just held out one, you see how well it fits the distribution. So basically, it can show if you consider the unbalanced effects. Actually, it improves in the accuracy of interpolating the distributions. And that's another example of EMT. They have like 5 time points, and actually using this approach, we can recover the stochastic dynamics, and also for the score matching, especially for the score matching, because log p in statistical physics, that's actually a way to visualize Waddington landscape, because the more stable is actually you have the larger probability, right? So basically, you can see the change of the stability of the cell states. At first, stem cell is very stable, but as time evolve, in the end, the differentiated states become more stable. So actually, there could all be inferred from the data, right? So basically, that's the idea of unbalanced and stochastic. That is called regularized unbalanced OT problem. So, so here I just want to show another work that is in progress. Is so far we only consider the independent particle system, but actually, what's also very interesting is how to infer the cell interactions from this the temporal sequencing data, right? So here the idea is actually in mathematical biology, people already have a way to model the interacting cells. That is called MV equation. So basically, in addition, you have RNA velocity and a diffusion and a growth. You also have an interacting kernel. That's called a non-local kernel. Here, phi is interacting potential. So of course, we could also generalize that to the non-gradient system, but let's just uh, do something for simplicity. We do start with something most elementary. We just assume there's interacting potential, but this potential is also neural network. We want to determine from the data, right? So we parameterize that, and the KXY is a kernel. For example, that's KN neighbor or something, or you have the prior knowledge. You could determine which cell can interact, and here interacting kernel just define how strong is interaction between different cells. So actually, this PDE also has a interacting particle system uh, interpretation, right? So basically, now you have the growth of the cell, that's a weighted particle, but you also have the interaction between different cells through the kernel, right? So here, idea is you parameterize this interacting potential, and you want to solve a optimal transport, minimize kinetic energy. So also use the previous neural OD solver to do that, to do the backpropagation. But here you could recover what is the interaction within the real data. So we also tried it on some
Sorry,
real,
like
yes,
have a question. So for the last
okay.
slide,
Uh-huh.
so how do you have the ground truth for the interactions between cell? So do you
Yeah,
have the
yeah.
ground truth first, and then you try to inject the ground truth as prior into the flow matching process?
Yes, that's a very great question. So basically, so I think for ground truth, it's more about the prior knowledge is about which cell can communicate with what cell. So you can use
Mm,
nowadays there are some tools like cell chat or like cell phone DB or like a Commot. They could
mhm.
infer which cell can communicate. So you build graph first. So actually, that's what this KXY mean. So whether it has an edge or not. But actually, in terms of dynamics, we don't know how strong this interaction will affect the transition dynamics of the cell. So that's here we use neural network to parameterize, to estimate this term from the data. So actually, for the ground truth, we make some simulation data. So they just have the ground truth interaction. We simulate this system, simulate the snapshots, and we just want to recover that. So actually, for the simulation data or synthetic data, it works quite well. You could recover the shape, at least the qualitatively, the shape of the interacting potential. Yes.
Just another
basically,
question. Thanks. So
yeah,
from the equation on the second line, I think this is the empirical version of the equation above, right? So you try
exactly,
to see the j not equal to I, which j is the interacting cells, and then you try
exactly.
to like maybe k is the interaction, and also w is the weight,
Yeah,
and then try to
yes,
learn the delta, like the network for the velocity for this
exactly.
interaction part. Okay,
Yes,
thanks.
here, yeah, B phi is different. So actually, the first line and the second line, they are equivalent because you just simulate the particle in the second line. Actually, we can prove the density, the distribution of the
Mhm.
cells will converge to this PDE. Yeah, so
Okay,
basically,
thanks.
yeah. But this way, you can do the back propagation using neural
Mhm.
OD solver to solve the problem. Yes, so basically that's the idea. So for example, that's the real data. So now we can not only estimate the growth, uh, estimate the steady states, but we can also estimate. So this is the learned interaction. For example, for the blood differentiation system, we found actually very interestingly is the stem cell interact dynamically with the differentiated states, and actually it has a pullback effects. It's just to inhibit the transition. We don't know why, but we can do some downstream analysis to identify some genes. So basically, it's an ongoing research. But I think another interesting observation is we do something in a cell line data. For example, that's the EMT induction data set. So actually from this data set, we found the interaction learned is very noisy. So actually, because that's an in vitro experiment, so the cell interaction is very small. They just do that in the plate well. So basically, we think this this potential term or this approach could somehow tell whether there exists the interaction or not, and tell the what is the role of the cell interaction in terms of the cell transition. But of course, there are a lot of to be done in terms of interpretability. So this is ongoing work. Another kind of ongoing work is we really want to do spatial, but actually doing the spatial data, there's another difficulty is the batch correction because or the slice alignment because the sample at different stage actually they come from different samples, biological samples or biological replicates, right? Because that's the limitation of the spatial temporal data or the spatial transcriptomics data. So at one hand, you need to correct the batch across 10 points. On the other hand, you want to estimate the underlying dynamics. So basically, we just want to do that in an iterative way. So of course, we also for simplicity, of course, later it could be relaxed. But at first, we assume this is there is a rigid body transformation between the geometry of different slices, just because of rotation or shift or something. But we could also be do some nonlinear transformation. But anyway, there we assume the batch effect happen in this parameterized form. And at the same time, so basically, you first parameterize a rigid body transformation between the slices. On the other hand, you also have the dynamical estimation, right? So here is a iterative strategy. You first identify what is the batch transformation. Then with the good transformation, we have the aligned slices, you do the dynamical estimation. But after you do some adjustment based on the dynamical estimation with the fitted data, perhaps we have a better way to align that, right? Because we correct some noises, so we can do that iteratively during the training process. So here is what happens is we have a ground truth simulation data, and we just collect some snapshots, and we just intentionally rotate that. For example, we only have 3 or 5 images Based on that, and each node, of course, also have the gene expression. It's just similar to the spatial transcriptomics data. And actually, this is what discovered or reconstructed from the algorithm. So actually, only with, for example, 5 slices, it could construct quite well. So of course, we have to assume the dynamics is not very complex, but for some simple or not too far measurements between consecutive measurements in time, it could even quantitatively recover the dynamics of this process, right? So here we just apply to the real data. For example, that's a Asacto brain regeneration data is actually you make a wand to the hemisphere of the brain of Asacto. What is fascinating about this Asacto is it could repair the wound even in the brain, right? It could regenerate. But here the issue is they only have 5 images, 5 snapshots, and at day 5, you can see the wound does not heal. There's still the gap here. But at day 10, the wound already healed. So what happens in the middle? Actually, it's a quite very expensive experiment, or even you want to see what happens in the middle. If you want to sequence more between, actually you need more biological replicates. Actually, they are not from the same one, right? So it's very hard to do that experimentally to see what happens in the middle. But here we could use algorithm to at least make an initial guess what happens in the middle. So we just use the algorithm to simulate that, reconstruct the dynamics, but we could discover a video based on that. So the story is the stem cell will first move to the wound, and then it will proliferate here a little bit, and then it will differentiate into the mature cell type, right? To finish the recovery process. And because and we can also find the proliferation rate at the wound, the cell growth at the wound is much larger than the control hemisphere, right? It also show the consistent biology here. And also another thing is here the deformation change or the rotation is very important. If you don't correct that, actually the dynamics is not as good. And what is a good thing about this dynamical generative model is, for example, you only have day 5, at day 10, they are already the differentiated cell. But with a dynamic OT model, you could just interplay what happens in the middle. What is the lineage here, right? So basically, that's one thing could be done. And also, what could be done is we could do some interpretability analysis, because we for each term here, it has a meaning. For example, what is spatial velocity? What is a gene regulation velocity? We could just take a partial derivative with respect to genes, and we can see what are the driver genes for the transition or the migration. Actually, we can do some pathway enrichment analysis to see whether this discovery makes sense. And we also recover dynamical gene regulatory network, because it just you have a nonlinear RNA velocity, and it takes the Jacobian of that term. That is how the genes are regulated, but changing with the time, right? Also for different location, different cell type, different space, we can construct a different gene regulatory network. But that's quite straightforward, because actually we have the equation. So that's quite straightforward downstream analysis to do. So also we could do the spatial, the 3D one. So basically here you only have 2 time points, because you want to, at one time point you want to do 3D, you need to cut them into slices. That's a very, very expensive experiment. So perhaps you only have 2 time points, right? So in this time, in this scenario, we could have more prior knowledge. For example, we can just specify which cell state transitions to which state. So in the end, at least empirically, we found we could reconstruct a quite reasonable process between 2 consecutive measurements, and we compare with other methods. For example, spatial or Moscow, actually it recovers more like at least the more coherent or consistent biological spatial structure from the reconstructed dynamics, right? So basically, I think now time is up. I will just make a summarize. Because here we just think through some dynamical equations, combine that with machine learning or generative models, we could do some virtual cell dynamics reconstructing task. And of course, here optimal transport or even more going to the depths that's about differential geometry. There are a lot of theoretical work that we could explore, and it also could be combined with foundation model. For example, nowadays we just do that in the PCA space, but what about we do that in a foundation model constraint space, or use that to better to make a prediction based on the foundation model. So basically, that's another very interesting top direction to explore. And in the end, biologically, we could also integrate that with other multimodality measurements. For example, now there are some live imaging data, but only track a few cells. But on the other hand, at very sparse time points, we could measure the whole genomics, right? The gene expression. So how to combine these two data? We have very few continuous data, and we have some snapshots. It could also be incorporated into this OT framework. So it just add more constraints about few genes, and we want to interpolate but impute dynamically of all genes. I think that's also very maybe an interesting topic to explore, right? So in the end, I just want to acknowledge my collaborators here, and also this is the references about the work that I showed today. So thank you again for this great opportunity, and I just want to hear more feedback, and also I want to have more discussion with you. Thank you very much.
Thank you, Professor Pager, for the wonderful presentation. I think at least I have a few questions, but we'll be opening up now a dedicated question section for the audience. So if you do have any questions, now is the moment to speak up or write them in the chat, please.
Okay.
I do have some question. The first one is that there are lots of mathematical details, and it's everything you showed is based on a strong mathematical background. But if we would like to try this method, how is the coding landscape today? Are there some libraries apart from Moscow from which we can quickly test the different methodology you've shown, or or is it something we would need to implement on our own?
Thank you very much for the question. So basically, now we have updated the package. I think that's in this preprint, or you can search name that's called Deep RUOT. So basically, that's based on the publication. So we have two versions of the package. So now it's written in the pipeline. So as long as there's a script, you just set that's called a Deep RUOT can serve on the GitHub. So basically, that's one possibility. Is you just have the hyperparameters, only a few, then you have the gene expression at different time, then you can automatically train that because that's all automatically done in the pipeline. So basically that's one possibility. The other thing, I think also that's a very, very great question, because we really want more biological researchers to use such algorithms. So actually we are now developing a package that is compatible with, for example, ScanPy or the ecosystem in the single cell analysis. So it's an ongoing work. So we really welcome more feedback based on that. So that's called a Settle Bridge. So basically that's also, I think soon will be online. The name is called, I think this one, that's called a Settle Bridge, but it's still under development, but we really want to make it soon to be published, and also is compatible with real problem. We have
great.
more to hear on that. Thank you. Thank you very much.
That's great. And I do have another quick question. One thing I also try to reconstruct trajectories. using optimal transport and flow matching.
Yeah.
But one specific concern I have is that I would like to observe these trajectories in a latent space,
Yes.
but not as you did, where you have a UMAP or latent representation of the cells
Yeah.
at different time.
Yeah.
Really, one dot being one trajectory. Here, one dot is one cell.
Exactly.
Is this
Yes.
something you have explored on how to embed this this time series actually in a latent space or not?
Okay, yeah. So basically, in the latent space, actually, we have done something that's a quite straightforward idea. Is you just train another uh, autoencoder, for example, or variation autoencoder. So just to train two things simultaneously. One is the dimension reduction or the latent space construction. So we have the reconstruction arrow. So basically, that's one loss. The other loss is dynamical loss. So just to train this either iteratively or simultaneously, so it could get a better visualization or embedding in the latent space that respect the dynamics. So basically, that's one thing we have already tried. So maybe I'm not sure whether it's the same as your idea, or
Okay.
another thing is nowadays if we want to trust the foundation model, we could also use the embedding of the foundation model directly to do
Yeah.
that. So basically, that's another. We have done that in some real applications, and for some data, it could work. Yeah, maybe work better than a PCA or UMAP.
Yeah.
Yes.
Okay. Thank you.
Yeah. Thank you. Thank you very much.
Maybe Dylan, do you want to go first?
Yeah, sure. I have a question. So I guess you were primarily focused on the spatial temporal mapping of cellular development. So a lot of focus was on single-cell RNA-seq or also spatial transcriptomic platforms.
Yes.
Do you think other types of modalities could also help? For example, there's like really nice cell lineage tracing technologies that could be super useful as grounding your work in biological priors. You've talked a lot of that, so I'm wondering what kind of data sets would be very firm foundations to add in as priors. I'm also thinking of epigenetics like ataxy, but I guess lineage tracing is very relevant here. So
yeah, that's a
thank
very,
you.
very great question. That's a very insightful question. So basically, this data set, especially this blood data set, is just from, I think it's from Alon Klein group. Basically, that's just the linear tracing data set. So they already have the linear tracing, but here we're just using linear tracing as a validation. But of course, we could make linear tracing as another constraint here. So basically, I think that's a very, very a great idea to do that. So hopefully that's a very good future direction to do it. Yeah, using the lineage tracing, the barcoding information,
Yeah,
or
and also
the reconstructed lineage. Yes.
And almost like when Philip was asking the previous question, the person who asked that question, Philip got me thinking about like a follow-up question to my own question, where if you're able to embed full trajectories in a latent space, right? Perhaps like you could import embeddings that already define full trajectories based on some ground truth information, like lineage tracing, for example.
Exactly.
And you import those in some dedicated trajectory-based embedding, as Philip said, where each trajectory would be like a point in some embedding space. So I'm saying this more as a follow-up thought or / question if you have other observations there.
Yeah, thank you very much. I think that's a very good strategy. So we haven't explored in terms of that, to make another high-level abstraction to make each trajectory at one point. We haven't done that, but definitely I think it's worth doing. So what we have done is still in the cell embedding space. We have tried some, more automatic or just a training-based way to do a dynamics informed embedding of the cells, but we haven't tried down in a in the trajectory space. but I think that definitely worth trying. And I think that's also relevant to the last point, also relevant to your previous question is, I think now for some live imaging data, or you can track some genes. That's a real trajectory, but only a few genes in the latent space or even in the real original gene space. So basically, that's also some very good constraints or biological constraints we could consider to combine that together with either optimum transport or flow matching map. Yeah, so basically that's a really great suggestion.
Thank you.
Thank you.
I have
you
a follow-up,
much.
but I'll stop for now.
Okay. Yeah, we could discuss later. Thank you very much. Yeah, thank you. It's a really, really insightful question, and indeed, yeah, there's a lot of things we could do in the future. Yes, based on this.
I'm sure.
Yeah, we can continue the discussion offline. You know.
Yes.
Let's go for that. So, in the
Yeah.
interest of time, since it's been a wonderful presentation, I will stop the recording here. Thank you, everybody, for coming to the 29th edition of the Multi-omics Reading Group.